Well, I would like to welcome you to Health Coach 7 webinar series on methylation. And today's topic is methylation and cancer. And I just wanted to let you know how exciting it is to be able to present this information on a very complicated disease process. And I'm just going to go through some uh, logistics here so that you can see some relationships in methylation and cancer. Cancer, of course, affects millions of people, multi-millions of people around the world due to various type of changes that may occur and maybe in genetic inheritance, of course, environmental exposures, various types of susceptibilities found in the genes. But it's not as simple as explaining, okay, well, I've just inherited cancer risk, so I'm going to get cancer. And the thing is that you should never ever throw in the towel, because the important thing is that if a person is diagnosed with cancer, they can always uh, do many things to actually create epigenetic intelligence that would minimize the susceptibilities that are actually seen in the genome, by actually creating good signals, turning off the bad signals, or silencing what they call tumor type cell expression or oncogenetic expression. So as you can see on the screen here, I actually did a general search on GeneCards, which is a very awesome database that you should consider being a public user on. You can look up any gene in the human genome. And as we talk about methylation here, I just did a DNA methylation search in cancer relationship, and it comes up with 3,013 different genes that are involved in the process that you might see that are affected by the changes that cancer brings in the DNA expression. Um, when you look at it, you can see there's various ones that are actually controlling DNA methyltransferases, which actually influence the cycle rate of DNA's replication. And of course, you got DNMT1 and DNMT3B, and then you can see some genes that you might recognize, like estrogen receptor and BRCA2 and BRCA1, which are very common genes that are actually studied. And then, of course, there's the ERB2s, which you probably would also recognize as Herceptin family of genetic controls. Various proliferating antigens. There's a whole lot. So if you actually went through all 121 pages of these 3,013 genes, you're going to find a significant amount of information on the genetic controls and crosstalk that goes on in the genetics. Now, what I want to do is I want to just show you that when we actually talk about implications in cancer genetics, we're actually talking about various types of cycling within the cells, to where the cells actually have a genetic control where the cycling becomes accelerated. So this schematic here is telling you what the cyclins and cyclin inhibitors do in the various what they call replication cycle of the cell. And in cancer cells, you can actually have hypermethylation or even hypomethylation occurring in the DNA that actually can trigger off accelerated cell cycling, which means that the cells actually reproduce themselves at a very fast rate. And then you can actually see that there's a transition that occurs in cell cycling. Now, when we talk about hypomethylation, we're talking about the taking away of methyl groups. When we talk about hypermethylation, we're talking about the addition of methyl groups. In both conditions, you will find cancer origins. And usually speaking, in tumor development, however, the literature points in the direction more of hypermethylation in relation to inflammatory controls, hypermethylation, and tumor development. So there's a lot of articles out there that actually express more hypermethylation than hypomethylation, but you can't rule out the fact that hypomethylation is not seen in cancers as well. Now, when we talk about cancer, we're talking about a lot of different factors, and things that you're seeing in this particular diagram is just basically telling you that if there is any one of these particular entities involved, you're going to find that each one of these categories are very relational to the survival and proliferation of cancer cells. Okay, so now let's look at this wheel really quick. We see self-sufficiency and growth signal, insensitivity to anti-growth signals. That simply means that if we've got environment where it's actually encouraging cell dysfunction to where the cells that were once normal in their DNA expression are now they've been hit by some kind of environmental factor, or they've actually caused there was some kind of a cause that actually created a shift in the nucleus of the cell, or the cell was hit by some harmful substance, or the susceptibilities are there. You're going to find that the regulation in these particular growth signals and growth factors may be altered. And so what winds up happening is there is then a shift into what they call a limitless replication, which actually causes the cells to begin to replicate at a high rate that are carrying a genetic message that necessarily is not necessarily healthy. So we call that, like, it could be a transition cell from a normal cell where it starts to bear antigens that are known as cancer-causing antigens. Now, the whole idea, you can see the interchange that's here when we talk about DNA damage and stress. I mean, that usually is the first place where methylation disturbances occur in the cell. Is the environment hits the DNA methylation cycle becomes altered, and then you actually have a progressive change in the ability to regulate the cell's reproductive rate. And then, of course, as you can see here, oxidative stress is a big influence on the protections of the cell. So anything pro-oxidative protections and protection from free radical injury, that's a big deal on these cells. And you're going to find that it actually causes a lot of mitotic stress, which means that if we find that these cells are agitated and that their DNA is actually hit, then what we wind up seeing is an accelerated rate of a bad message of these cells. Then they replicate in the exponential range where, one becomes 10,000, 10,000 becomes 20,000, and before you know it, you've got, some multi-million to billion cells that are affected. And remember, you got 60 trillion cells in the body, so these cancer cells can actually move that quickly. So you can see metabolic stresses, proteotoxic stresses, which means that proteins are then becoming developed that shouldn't be. That's when you start getting antigens, cancer-related antigens that are expressed on the cell surfaces, because that's the proteins then that are being made are abnormal proteins because the DNA messaging system on the genes has actually been hit. And then you get, the cells themselves develop intelligence to where they evade the immune surveillance of the body. So that means that the immune system is not able to detect these cells very, very well. And then you can see the relationship in the environment of how the cells love to respond and exist in a low pH environment. So that means in an acidic pH. They love to be sustained by a low oxygen environment so that the cell's survival rate is high with low oxygen, acidic pH. So you can see where we're going with this. If we create the environment to where the cells will not survive, where we actually alkalinize the pH, we expose the cells to oxygen, we deprive them of sugar, we actually control the exposure rate that might hit healthy cells and create more harm on those cells. We actually create more immune strengthening to actually survey these cells appropriately, raising up the natural killer cells and stem cells that are defending them, drop the inflammatory rate down, et cetera, et cetera. So you can see that this, what they call cancer survival wheel, is a very, very important process in being able to see how these cancer cells survive and proliferate in the environment. And ultimately is when you have a normal cell, the normal cell is clean. When you have a transition cell is kind of like between a normal cell and a oncogenetic cell, fully oncomutated cell. the transition cell bears some antigens that are abnormal antigens that are considered to cancer risk antigens, and then the fully mutated cell actually bears a complete schema of what they call tumor-specific antigens that you won't see on normal cells. And when it talks about aneuploidy, what that simply means is that if you looked at those cells under the microscope, you would actually find that the nucleus of those cells are misshapen, and a normal nucleus has a nice intact round symmetry of the nucleus, very predictable, showing that the DNA in the nucleus is not going crazy and replicating as, breaks in the double strand, to where then it wraps around, and a double stranded DNA is just a single strand of a mess. That's what creates the misshapen nucleus, is because DNA has just gone wild in the nucleus, and it just keeps on coiling and coiling and coiling and then the reliable messages that once were in the cell are no longer there, so the cell then proliferates. And that DNA methylation cycle is a big part of that. So hyper and hypomethylation are all part of that whole process. So as you can see, when we talk about genome stability, so when we say susceptibilities and sensitivities in cancer, it's very important to realize that in cancer, the epigenetic mechanisms are hit, and so that means that you have a lot of negative, what they call mutations, that are occurring, that actually create an expression of what they call upregulated products of highly genetic mutation messaging genes. And what you have then is alteration in proteins, alteration in enzyme control. You actually have, from what you once had as normal cells that were moving, through the process of a hit instability in the genome. And because the epigenetic signals are encouraged to favor unfavorable signals, then you actually have disturbances that are occurring that actually are creating a proliferation in the cells that I just showed you that last diagram, an exponential amount of cells that are being produced with bad nucleus and DNA in that nucleus. So and that actually causes the cancer to continue to develop and proliferate. So as you can see here, what this is telling you is that DNA methylation is very much part of the coding process of keeping an intact normal structure of the cells, so that they don't transition into a fully oncomutated cell. Okay, so mutations, that's how that happens. As you can see, this again too, this is just a basic cell cycling, where it's showing you DNA methylation involved in silencing specific gene controls that would actually stop certain aspects of the cycle of the cell, and it would arrest the ability of that cell to continue to replicate. So this is basically showing you how you can pretty much influence certain type of gene controls to maybe arrest certain cell cycles at a certain point, to discourage the formation of future progeny of mutated cells. So that's DNA methylation is involved in that process. And as you can see, there's no way that we can get away from DNA methylation's influence on cancer cell progression. And then here you can see a normal cell has a normal lattice of DNA showing a very predictable pattern. Here you see where a number of epigenetic signaling factors are involved, to where there's a whole lot of influence on the DNA, causing methylation disturbance. It's a very simple diagram to explain that. It's very, very important to kind of take this as a whole picture, okay? Now, here we see, this is a very good diagram and showing with relationship to colon cancer and DNA hypomethylation patterns in oncogene activation. Okay, so here's where hypomethylation is involved, and here you can see that decreased dietary folate intake is actually a trigger for relational movement on the DNA methylation to cause a hypomethylation disturbance, and then you have what they call faulty DNA replication. So normal DNA replication is not happening, faulty DNA is happening, and that's when we start having DNA repair breaks, chromosomal involvement, when the genes are actually unhappy. And so then you get the bad signals that actually occur from that. Now, there were a couple of questions that actually were really good on, one is BRCA1 and BRCA2 on my 23andMe results, and can that be used as a diagnostic criteria? No, you can't use BRCA1 and BRCA2 on 23andMe as diagnostic criteria. Yes, the BRCA1, BRCA2, some SNPs are on BRCA1 and BRCA2 that are listed on 23andMe. However, one of the things you need to remember is that just because BRCA1 and BRCA2 comes up as a positive on a typing doesn't necessarily mean that you're going to have a full expression of that particular cancer cell type that bears that mutation. And sometimes cancer cells can shift in their ability to express. So, like in other words, on HER2/neu and women who were trying to qualify for Herceptin, initially these women may have been tested and they were negative on Herceptin, and then through a stage of traditional conventional treatment, received chemotherapy, and then they were tested again on HER2/neu. And what was discovered is that when they were initially negative, and then after chemo treatment they became positive. So what winds up happening there is that the new cancer mutation cells actually would actually bear the image of HER2/neu receptor on the cells. So there was actually a transition that occurred to where those cells were now expressing HER2/neu. And then the reverse has occurred to where initially women who were expressing HER2/neu then became negative. So remember, various type of hormonal controls in breast cancer and other cancers, like estrogen receptors, progesterone receptors, HER2/neu, various types of cell cycle growth regulators, tumor necrosis factor. There's a bunch of different upregulated mutated gene products that are bearing on the cell surfaces. So this is a good image here to show you hypomethylation's involvement. So that means that if you have a decreased dietary folate increase, you're going to increase your risk for DNA hypomethylation that would influence colorectal cancer by causing faulty DNA synthesis. And a lot of the times colon cancer recipients are receiving a folate analog or a type of a folate like leucovorin as part of their conventional treatment that would actually assist with better DNA management. So it's really important to see that. Now here's the other thing: DNA methylation cycles in the ability to give dependable movement in keeping silenced genes, the genes that are responsible for cancer-causing cells. This is actually just showing you the loop of gene silencing. A certain amount of gene silencing is necessary to stop the recruitment of a certain type of proteins that might actually influence the proliferation of the cancer cells. And this is just showing you this one H3K27Me3 dependent silencing mechanism to show you how it's influencing the cancer-specific tissues. Okay. When we actually look at these type of charts, we just don't want to go nutso, but I want to just show you that this is a very good chart. I don't want to create more confusion, but the thing is that look at this chart, because this is actually the cancer immunoediting movement showing you how cancer cells proliferate and what the immune system is supposed to be doing to try to create a defense movement against the cancer so that these cancer cells do not escape and that they are eliminated through that process. Now, you've probably heard of cases where there have been progressive spontaneous degeneration of cancer cells, and that is actually that's in other words of where cancers have spontaneously disappeared. Well, mechanisms it's actually called oncogenic. It's an oncogenic process where it actually is a reversal process of the cancer cell progression. So it's very important to know that just like the cancer cells are moving and trying to proliferate really fast, all of a sudden there could be a cascade of a process called oncogenic addiction, to where then the cells become susceptible, and if the one cancer cell line dies, there's a cascade of self-destruct to where then the cancer cells begin to disintegrate at a very fast rate. And this happens in many cases. And I've treated cases over the last 20 years where spontaneous regression has occurred because the immune system has kicked in and eliminated those cancer cells. And the methylation cycle of DNA is very much involved in that. So as you can see, this is a very, very comprehensive subject with a lot of bells and whistles that are involved in the process of keeping normal process in the body and preventing cancer. And of course, don't believe for 1 minute that if a person is diagnosed with cancer, that their body is not trying to develop a more efficient way to defend. And so a lot of the times we wind up rushing into therapies that we think may be helpful, but we certainly don't want to be destroying the immune recognition software, because I'll tell you what, some of the conventional therapies do that precisely. They wipe out the immune system's ability for future recognition, thereby creating a higher risk for recurrence and maybe even resistance. So I wanted to present all this to you. Thank you for attending today. I think it was great that you came today on seeing cancer and DNA methylation. And thank you for asking those questions. I think the important thing is remember that you can create epigenetic intelligence to address what you would see as a process that's not acceptable in the human body and cancer causing such as like this. And it's very important when you're actually working up your health to always look at the whole picture. And here at Health Coach 7, that's what we do. We look at the genetics and epigenetics and try to figure out ways to improve the signaling and help with the movement of better, vibrant health. And thank you again. We'll talk next time on the methylation series. Look forward to our next topic. And then also a new program we're developing. It's called Gene Crosstalk, Gene by Gene. We're going to actually talk about how genes communicate with other genes, and you're going to actually get to see how that process happens. I think I already showed you the chart on the gene cards there. I think there was one more. I'm looking here. Oh, yeah, here's a cancer cell. I just wanted to show you this before I leave. There's a normal cell. Here's where you're seeing an abnormal cancer cell. So you can see the nucleus is not good, and then it's bearing a lot of mutated products on the cell surface. So that's one of the things that, you can get these slides here, by the way, on this here. So here's another very good article that you may want to look at, and it's actually called DNA and Methylation and Cancer. And I found this article to be very, very helpful in explaining the patterns that are involved in cancer development. And then, so feel free to, look at that when you have a chance. All right, I think that pretty much sums up what we're doing, and I look forward to seeing you next time. Thank you for your attentiveness on this DNA methylation and cancer, and we'll talk next week. Bye-bye.